Hello, everyone. Today, I'd like to take you on a journey inside your own body into one of the most complex and fascinating biological systems we know, the human immune system. Every day, the immune system would make hundreds of billions of red cells organized as an army, with soldiers, captains, intelligence officers, heavy weapons, and crazy suicide bombers. It's not just some sort of abstract entity, but actually, your immune system is you. Your biology system protecting you from the billions of microorganisms that wanted to consume you, and from even your own perverted cell that turned into cancer. In fact, each of us has 1 to 5 cells undergo cancerous changes every single day, and every day your immune system quietly eliminates them before we ever know. In the next few minutes, I will walk you through how the system works, connected to a lab experiment that we've done on bacteria transformation in our BIOL110 lecture, and close with a real-world application that affects millions of people daily. So first, let's move on to the background introduction about the two lines of defense. The immune system operates in two major layers that work in sequence. The first is the innate immunity. Every single animal on Earth has this. It's the ancient universal defense. It activates immediately upon infection, within seconds to minutes. Rather than recognizing specific threats, it detects broad patterns shared by many pathogens, including bacterial cell walls, flagellin protein on bacterial surfaces, double-stranded RNA from viruses. It does this through receptors called toll-like receptors, or TLRs. The tools of innate immunity include physical barriers like skin, mucous membrane, antimicrobial proteins, and the inflammatory response. The second layer is adaptive immunity, exclusive to vertebrates. It's slower, sometimes taking several days to fully deploy, but it is extraordinarily precise. It doesn't recognize broad patterns, but rather specific molecular signatures on specific pathogens. It does this through B cells and T cells, each carrying unique antigen receptors. Most importantly, adaptive immunity has memory. Once it learns a pathogen, it remembers it sometimes for life. These two systems don't operate in isolation. Innate immunity fires first and buys critical time. And importantly, it is the innate immune response, specifically dendritic cells, that activates the adaptive response. Think of innate immunity as emergency first responders, and you can think adaptive immunity as the specialized forces that arrive later with exactly the right weapons. So now let's look more closely at the key players of the innate immune response. The first to arrive at any infection site are macrophages, literally big eaters. These large cells extend projection like the arms of an octopus. They grab bacteria and engulf them whole through phagocytosis. A single macrophage can consume up to 100 bacteria before it's exhausted. But they are not just like killers. They also release chemical alarm signals called cytokines that recruit reinforcements, and later in the response, helper T cells can actually reinvigorate a tired macrophage, giving it a burst of renewed energy to keep fighting. Next come neutrophils, and these are the true foot soldiers. They are short-lived, intense cells that exist purely to destroy. They vomit toxic chemicals onto bacteria, engulf and digest them. And some neutrophils go even further, that they express their own self-destruct button and exploit, releasing white nets made from their own DNA filled with an antibacterial compound to trap and kill pathogen even in death. This is how devoted the cells are to protect you. Then we have natural killer cells, circulating in the blood hunting for cells that look abnormal. Once there are cells infected by viruses or cells that have become cancerous. When a natural killer cell finds one, it might trigger the target cell to undergo apoptosis, a controlled, programmed self-destruction, and stopping the threat from spreading before it can do more damage. And finally, the dendritic cell, which is acting as the intelligence officers in this battle. While the battle rages, the dendritic cell quietly collects fragments of the invading pathogen, then leaves the battlefield and travels through the lymphatic network to the lymph nodes. There, it will activate the adaptive immune response. It is the essential bridge between the fast, broad innate response and the slow, precise adaptive response. So, how does the adaptive immune response actually unfold? It could be break into 4 steps. The step 1 is the antigen presentation, that the dendritic cell arrives at the lymph node carrying pathogen fragments called antigen, displayed on its surface, bound to proteins called class II MHC moleculars. This is essentially the immune system's wanted poster, showing every T cell it means exactly what it's looking for. Step 2 is the helper T cell activation. Among the billions of T cells in your body, each carries a unique receptor shaped by random genetic rearrangement. The dendritic cell moves through the lymph node, making contact with T cell after T cell. Most don't react because the shape doesn't fit, but one will do. That T cell's receptor matches the antigen perfectly. Cytokine signals are exchanged, and that helps the helper T cell become activated. This is the critical moment that the immune system has found its weapon. Step 3 is the clonal selection. The activated helper T cell begins cloning itself. 1 becomes 2, and 2 becomes more, until there are thousands of them. These clones split into 2 groups. One group rushes back to the infection site to reinvigorate macrophages with chemical signals, while the other group activates B cell and cytotoxic T cell to begin the full adaptive assault. The step 4 is antibody projection and memory. The activated B cell differentiate into plasma cell, which is the antibody factories, and each would secrete up to 2,000 antibody per second. These antibodies coat bacteria, neutralize viruses, clump pathogens together, and mark them for destruction. After the battle is won, most effector cells die back, but a small population of B and T memory cells remain, sometimes for the rest of your life, ready to mount a dramatically faster response if the same threat ever returns. This is the biological basis of vaccination. So now let me connect this directly to one of our lab is about bacterial transformation experiment. In this lab, we introduced the PUC19 plasmid into competent E. coli DH5 alpha cells using the heat shock method. The PUC19 plasmid carries 2 key genes. The first is the AMPR gene, which encodes beta-lactamase, an enzyme that degrades antibiotic ampicillin, and the LAC-Z gene, which produces beta-galactosidase and turns colonies blue in the presence of exgo. We plated transformed and untransformed cell onto 2 types of media, selected plates containing ampicillin and non-selected plates without it. The result was striking. On the ampicillin plates, only the transformed cells survived and formed blue colonies. On the non-selective plates, untransformed cells proliferated freely and outnumbered transformed ones, forming dense white colonies. And on the ampicillin place with no plasmid, no growth at all. So what does it mean for immunology? The connection is a concept of selection pressure. Ampicillin on those plates function exactly like the immune system within our body. It killed everything that lacked the resistant gene and allowed only the resistant survivors to reproduce. This is precisely how antibiotic-resistant bacteria evolve in clinical settings. Bacteria that occur resistance genes through transformation or mutation gain a massive survival advantage under antibiotic pressure. And this lab also illustrates horizontal gene transfer, which is about the bacteria acquiring new gene from their environment without reproducing. This is one of the primary mechanisms by which resistance spreads across bacterial populations. The immune system, powerful as it is, evolved to recognize and destroy specific pathogens. When those pathogens rapidly occur new genetic traits through horizontal transfer, they become a moving target. And the adaptive immune response, which takes days to deploy, cannot keep pace. For our real-world application, let's talk about a medicine that is widely used, antihistamines. So allergies occur when the adaptive immune system makes a specific kind of mistake. A completely harmless substance, such as pollen or certain foods, might get mistakenly identified as a threat. The adaptive immune system produces IgE antibodies against these allergens, and those antibodies coat the surface of mast cells, which are innate immune cells found in connective tissue throughout the body. The next time that allergen antibody it binds to IgE antibodies on the mast cells, this cross-linking is a signal. The mast cells rapidly degranulate, releasing histamine, and histamine then binds to H1 receptors on blood vessels and surrounding tissues, triggering vasodilation, swelling, itching, and mucus secretion, the classic allergy symptoms. Antihistamines are competitive antagonists. They bind to the same H1 receptor that histamine targets, but they don't activate it. They simply occupy the receptor, physically blocking histamine from binding. The mast cells still release histamine, while the histamine doesn't stop the upstream step, but the signal has nowhere to land. More importantly, antihistamine do not suppress this broader immune system. B-cell antibody production continues, and also the T-cell response continues. The body remains fully capable of fighting actual infections. The intervention is precisely targeted at one receptor in one downstream signaling step. This is a perfect example of a principle we keep seeing, that the immune system is not a single switch that you could turn on or off. It is a network of discrete targeted signals, and the best strategy works by interrupting one specific signal with minimal disruption to everything else. So, in general, your immune system is doing all of these right now, silently, continuously on your behalf. And I hope this presentation would give you a clear window into the extraordinary biology keeping you alive every day. Thank you for your listening.